Welcome to The Explainer. Today we are setting off on an absolutely incredible journey from the macro right down to the micro, diving straight into the fundamental mechanics of life itself. I mean, look down at your hands right now. Take a deep breath. Consider for just a second that you are literally a walking, talking universe made up of trillions of tiny, living compartments. We aren't just looking at standard biology today. We're exploring the microscopic architecture that makes your very existence possible.
But before we look inward, we kind of have to look back, and I mean way back. It poses this really fundamental mystery. Where does a cell actually come from? To answer that, we've got to rewind the clock about 3.5 billion years, which is long before dinosaurs, long before plants, straight back to the raw, completely volatile environment of the early Earth. Okay, let's dive into this origin story because it's wild. It's pretty widely accepted by scientists that life began in water. But we are not talking about a calm, gentle ocean here. Think extreme, boiling, totally hostile environments. For instance, researchers studied the hot springs of Puga Valley in India, where temperatures just hover right near the boiling point, even in freezing climates. And guess what? They found mostly single-celled, heat-loving bacteria called thermophiles just thriving there. What scientists figured out was that calcium carbonate formed rapidly around these springs. These mineral deposits likely shielded early organic molecules from deadly radiation and, incredibly, helped form the very first protective membranes. Basically, the initial boundaries that defined the very first cells. Now, these microscopic entities have been around for billions of years, right? But for almost all of human history, we had absolutely zero idea they even existed. Why? Well, purely because of our own biological limitations. Think about it like this. If you draw two tiny dots on a piece of paper and bring them closer and closer together, your unaided eye can only distinguish them as separate objects down to a distance of about 0.1 millimeters. That is our human limit of resolution. Anything smaller than that just blurs into a single point. So, to actually see the building blocks of life, we had to invent technology that could break the ceiling of our own visual limits, taking us from a tenth of a millimeter all the way down to the nanometer scale. The tool that finally shattered that barrier for us was the light microscope. Back in 1665, a scientist named Robert Hooke designed his own custom version, and it was capable of magnifying objects about 200 to 300 times. By placing just a super thin slice of cork under visible light, he observed these tiny, box-like compartments. He thought they looked like small rooms, so he decided to name them cells. That one single observation completely changed our understanding of life forever. Now, light microscopes are fantastic. We still use them in school labs all the time today, but modern science needed to go deeper. So enter the electron microscope. Instead of using visible light and glass lenses, this massive, futuristic-looking instrument fires a literal beam of electrons to produce highly magnified images. This tech allows us to see structures at the nanometer scale. To put that in perspective, a nanometer is 1/1000th of a meter. We went from just seeing the rough, blurry outline of a cell to seeing the mind-blowing, intricate machinery actually operating inside it. So you might be wondering, how do scientists actually know the real physical size of what they're looking at under a lens? It's basically a clever little workaround. You simply place a transparent millimeter ruler under the microscope, measure the diameter of your whole field of view, and convert that number into micrometers. Then you swap the ruler out for your actual slide, let's say an onion peel, and just count how many cells fit across that exact same line. Divide the total space by the number of cells, and boom, you have your physical size. Let's do some quick math just to see how that works. If your field of view is, say, 5,000 micrometers wide, and 25 onion cells stretch all the way across it, then a single onion cell is about 200 micrometers wide. Add 100x magnification from your microscope on top of that, and that tiny building block suddenly appears 100 times larger to your eye. Pretty neat, right? All right, let's step back for a second and see how all this builds our understanding of complex life. We've established that the cell is the fundamental unit, but life is highly organized. It's like a giant puzzle. A single cell is powerful, sure, but a group of similar cells working together forms a tissue. Then different tissues organize to form an organ, like your heart or your lungs. And several organs work together to form an entire organ system, like your respiratory system. From a microscopic 200 micrometer building block all the way up to a living, breathing human being, it's just a perfect hierarchy of life.
Which brings us to section 1, the universal gatekeeper, this cell membrane. For these trillions of cells in your body to work as a unified system, they absolutely have to be able to talk to each other and interact with their environment. They rely on this boundary that is just 7 to 10 nanometers thick. Scientists call the way this ultra-thin barrier works the fluid mosaic model. It's made of a lipid bilayer, which is essentially just 2 layers of fat molecules. The outer heads love water, while the inner tails completely repel it. But here's the cool part. It's not a rigid, solid wall. It's highly fluid. The molecules can actually move sideways, flip around, and rotate. And embedded throughout this moving sea of fats are various proteins, arranged kind of like moving tiles in a mosaic. These proteins act like strict bouncers at an exclusive club, making the membrane selectively permeable. That basically means they actively choose exactly which substances get in and which get kicked to the curb.
To really grasp how this selectively permeable membrane responds to its environment, let's look at some simple kitchen science that was outlined in the source material. If you take 2 identical pieces of potato, place one in plain tap water and the other in a 20% salt or sugar solution, and just wait an hour, the physical effects are actually dramatic. The potato piece in plain water physically swells up, but the potato piece in the concentrated salt solution noticeably shrinks.
The reason this happens comes down to a really simple battle of concentration. It is all about the solutes inside the cell versus outside the cell. If the outside liquid has fewer solutes than the inside, which we call a hypotonic solution, like plain water, water literally rushes into the cell, making it swell up like a balloon. If the concentrations are perfectly equal, an isotonic solution, the cell stays exactly the same size. No big deal. But if the outside liquid is highly concentrated with salt, a hypertonic solution, water is aggressively sucked out of the cell, causing it to shrivel and shrink.
But this fluid dynamic behaves slightly differently depending on the exact type of cell we're looking at. Just notice the stark visual contrast here. On one side, we have plant cells from an onion peel. They are super box-shaped, rigid, and regularly arranged, kind of like bricks in a wall. On the other side, we have human cheek cells, animal cells. They are totally irregular in shape, kind of blobby and scattered. So why such a drastic difference in their cellular architecture?
Well, the answer lies in an additional layer of armor. Plant, fungi, and bacteria cells have this tough, rigid outer layer called the cell wall that surrounds their cell membrane. This gives them that structural, brick-like shape and actually protects them from bursting if they happen to take in way too much water. Animal cells, however, like the cells inside our own cheeks, do not have a cell wall at all. They only have that flexible cell membrane. And that is exactly why animal cells are so vulnerable to shrinking and losing their shape when they're exposed to highly concentrated solutions.
So, from ancient boiling hot springs to the fluid mosaics operating inside you this very second, the cell is just a complete masterclass in biological engineering. The fact that an invisible boundary just nanometers thick dictates whether a cell swells, shrinks, survives, or dies is, well, it's nothing short of miraculous. And it leaves us with a pretty fascinating thought to ponder. If these microscopic boundaries dictate your entire physical form, what unseen cellular interactions are shaping you right now, in this very moment? Thanks so much for joining me on this explainer. Keep wondering, keep observing, and I'll catch you on the next one.